I will pass the floor to the coordinator of the secreted project. This is Dr. Manuel Salvador de Lara from IDENER, and he's going to give us also a presentation of the secreted main objectives and scope. So, dear Manuel, we see your presentation. Great. So,
yes, you can see it, right?
Yes, yes, we can see it. Great. Yes.
Okay, perfect. Shall I start?
Yes, of course, of course.
Okay, then. So thank you.
Thank you.
So much, Costas, for organizing this amazing clustering activity. And thank you so much for all the attendees to assist to these clustering activities. I'm really grateful of participating on this. And let's start. So Secreted Project stands for Sustainable Exploitation of Bio-based Compounds Revealed and Engineered from Natural Sources. So it's like a double meaning word in the acronym because we are speaking about material secretions. Those are quite easy, let's say, to isolate and purify if compared with other compounds. And so I'm going to give you a brief overview of the background of the molecules that we have selected as secretions from bacteria that are biosurfactants and siderophores. I will give an overview of the consortium and the project and the partners that belong to the consortium. I will briefly explain the general objectives of the project, and I will explain also the concept and approach and the impact, expected impact of our project. So, as I said, we are focused on biosurfactants and siderophores, and surfactants are a very diverse group of lipid-like molecules which have an amphiphilic character containing both hydrophilic and hydrophobic domains in one molecule. This confers this family of molecules with two major abilities. They can lower the interfacial tension, allowing, for example, to solubilize hydrophobic substances, that is mainly the action of what we know as a detergent. And they can organize also themselves in complex supramolecular structures, such as micelle, lysozymes, or microemulsions, which is really interesting for the nanoencapsulation industry. So biosurfactants, which are the surfactants that are mainly produced by microorganisms and also other taxa, have also reported important bioactivities, including antimicrobial, antifungal, anti-inflammatory, and anti-tumor, leading that to bed a lot of applications based on their amphiphilic character as the nanotechnology and their use of possible pharmaceuticals because of these bioactivities. They are also used in based on their capacity to solubilize hydrophobic substances in bioremediation of oil dispersal for oil recovery also. And one of the abilities that is also considered is that biosurfactants have also chelating properties, being in some cases hard to distinguish from amphiphilic siderophores, which are the marine siderophores, that they also have this amphiphilic character containing both hydrophilic and hydrophobic domains. In the case of siderophores, we reminded that those are chelating molecules which bacteria secretes to capture iron from surrounding environment. And they have also, because of this chelating character, applications in health. They have been used to diminish the iron overload after a blood transfusion. They have also an anti-tumor role because they starve cancer cells by depleting the nutrients, in this case, iron. And they have applications in the green industry because they favor plant and microsphere growth against plant-associated pathogens. And because of the chelating ability, they have a role in the bioremediation by immobilizing heavy metals. So this is why mostly biosurfactants is one of the 5 topics with significant potential for industrial biotechnology in the European Union. but there are still some technical and economical obstacles to establish biosurfactants in market. And those are the higher production costs compared to their chemically derived counterparts, and their limitations in their chemical diversity. So to overcome these two major difficulties, Secreted proposes to expand the range of the chemical diversity of these compounds by analyzing the individual steps that conforms the biochemical pathways in charge of their synthesis. So those it would be a matter of discovering the biosynthetic gene clusters that are in charge of these compounds and isolating and standardizing the genetic elements that constitute them to be further combined and introduced in proper amenable industrial strains. So we have inspected available databases to obtain all genetic and biochemical information related to siderophores and biosurfactants synthesis and the information regarding molecular structure, molecular properties, and to perform new tailor-made compounds based on the combinations of already known genetic elements to further insert them into suitable industrial strains. So in the meantime, we have within Security Consortium available microbial collections that will be screened for such compounds. Those with proved novelty will be reverse engineered to fit this design chain and will refine and complete our database completion. So the endless space of possible combinations is going to be constrained by both the availability of a proper industrial strain that can work as a host for the synthesis and new designs, which means basically what are economically feasible in terms of the yield production, yields, and suitability for the use in a typical process engineer. And also, this endless space of combinations is going to be constrained by the secretive users, end users' needs that are basically giving some properties they are looking for to tailor cosmetics, household care, nutrition, agrochemical industry, genetic material delivery, anti-tumor, and bactericidal agents. Altogether, we can resume this project by saying that we have a family of compounds which possess a high variety of applications. And based on their chemical nature, they can be easily modified. So, for example, just by interconverting the hydrophilic and hydrophobic domains. And we are going to deepen into the genetic and biochemical knowledge that is available in both already databases and hidden in our collections. What we are going to use is this knowledge to design tailor-made molecules to satisfy our industry requirements. So Secretive, the consortium, is basically composed by 15 partners that are belonging to 8 European countries and comprises 4 universities, 6 small medium enterprises, 2 large enterprises, and 2 research transfer organizations. But what is really important in Secretive is that we have stakeholders of our proposed technology that works also as research and development and engineer partners. So basically presenting our consortium, we have the coordinator, that is IDENER, that is in charge of all the computational approach, that is basically all the chemoinformatics, model assisting strain design, pathway design, process modeling. We have also PharmaMar, that is in charge of screening their collections. They will also inspect their collection for anti-tumor activities within the spectra of our molecules. We have Acuplex Diagnostics, whose core expertise in siderophore is going to complete our approach, and they will give expertise in the purification and chemical modification for the further applications. We have Matis, we have a large experience in previous European projects, also screening their microbial collections, whose physiology is they are experts in the thermophilic physiology. We have a Station Zoologique Anton Dorn, also with a large experience in inspecting microbial strains from deep sea and Arctic and Antarctic ocean. And they will be focused on the cold adaptive physiologies for the durable production of these compounds. We have the University of Athens, who are leaders in chemical investigations, and they have also a large experience in these kind of approaches. The University of Tübingen, who's have a large experience also in filling the roles of biosynthetic gene cluster, because those are one of the founders of the Profilic platform anti-smash. Blue Synergy, who is in charge of the process assessment, life cycle analysis, socioeconomic, techno-economic, that is very important in our project. Bio-based Zero Pilot Plant, who have a large experience in upscaling the production of biosurfactants in previous European projects. Sphera Technologies, that are experts on nanoencapsulation formulations, and they will be in charge of assessing the convenience of our new formulations for the use in nanotechnologies. Imperial College of London, who are one of the most important synthetic biology institutes in the world, and they will be in charge of developing our new strains. University of Seville, who are experts on allophilic physiology, and they will complement our expert group with covering the allophilic physiology that is related to marine bacteria. And Lund, that are also experts in biosurfactants. They are also experts in upscaling and optimization of lab-scale fermentation. Exelisis, that is present Costas, who is our dissemination communication, and Silentis, who will inspect the convenience of these formulations for genetic material delivery, especially in RNA interference treatments. So overall, our objectives are to screen secreting microbial collections for biosurfactants and siderophores to infer novel biosynthetic gene clusters, and to construct an integrated genomic and chemical database, which is going to contain all information that is being collected along all these years about siderophores, biosurfactants, other molecular properties, all the molecular features, the chemical structures, physicochemical and functional properties. That is going to be further completed with the information that is going to be collected from the microbial collections. Another objective is to produce and validate a mix and match modeling pipeline in order to develop new formulations from the combination of the genetic elements that form part of the biosynthetic gene clusters, and proceed to optimize these heterologous producers with metabolic engineering. So we also have as an objective to characterize the compounds that we produce and we discover, and develop and validate a whole integrated mathematical model for all the production process, from the molecular design to the process engineering. Finally, of course, we will demonstrate the proposed solutions at a high technological level by upscaling a pilot plant, selected bioprocesses. And of course, as is necessary, we will assess that all the process is sustainable from an environmental, economical, and it will be also accepted by the social perspective. And of course, everything has to be disseminated. So the concept of security relies on the reutilizing previously collected microbial collections. It is a matter of not proceeding with new screening campaigns. So at first, we will utilize all collected microbes in these so-known microbial collections. And that is the action of work package 2. And it will be fully dedicated to data analysis, integration, and so will be dedicated to assess and refine the screening methods that will be applied in the extensive microbial collections provided by PharmaMar, Estación Solvit and Tondon, and Matis. University of Seville will extend this screening incorporating allophilic bacteria from public bacterial collections. And the major objective of this is to collect all possible hits and characterize the genetic elements that are in charge of their synthesis to be further combined for the uses in these industrial applications. As I said, the security is fully oriented to the industrial applications. So the industrial end users of security, they have given to us, and they will refine their specifications, and that will run machine learning approaches to infer the properties of not known to date molecular designs. So that will require the analysis and integration of already available information and their completion with quantitative structure activity relationship models, and also their individualized inferral of the genetic elements that are in charge of the different parts of the molecules. We will also be in charge of the evaluations of the different microbial strains and their ability to produce these compounds from raw feedstocks by using genome scale metabolic models. So as a brief summary of what we have collected so far in our approach, we have collected from the public available databases more than 1,900 unique compounds, more than 1,200 biosurfactants, and more than 700 siderophores. Of them, more than 1,700, they have defined molecular structures, which permitted the calculation of molecular descriptors for the inference of different properties that are envisioned in secreted. That those are quite a lot of 1,600 molecular descriptors in each molecule. We have also grouped the secreted biochemical space based on their similarity, in their molecular similarity, in more than 360 molecular families. And the availability of the calculations of these molecular descriptors has facilitated the inference of a lot of the interesting properties that are envisioned in secreted, and the process is ongoing, as you can see. So with the starting data, we have trained machine learning models that they have inferred with a high prediction accuracy these features in a wider aspect to be valued in our purpose. We have also collected the information regarding their origin, the optimal microbial producers, the unique microbial taxa that are associated to the synthesis of these compounds. And this information is going to be used to infer the biosynthetic gene cluster that are encoded in this bacteria. So as a further step in this DBTL cycle, design, build test and learn cycle. The work package 4 its aim is to construct rational engineer strains with the capacity of reproducing new chemical designs. So those will build the designs that are envisioned in based on our computational approaches. And to this end, different tasks will be developed in parallel. So we will evaluate the convenient production chassis. We will assess the functionality of the combinatorial gene clusters. And we will engineer the strains with the capacity of reproducing new chemical designs. Everything will be constructed through the further validation. The test phase consists in the lab scale fermentation chemical characterization. So this work package oversees assessing and assuring the novelty of convenience of discovered and engineered compounds. And in this work package, we will collaborate to establish lab scale protocols amenable for scaling up cultivation and purification of target products when required. So this is also evaluated from our partner from Bio pilot plan that will be in charge of assuring that any single process that passes this phase of the project is going to be able to be scaled up to an industrial scale. And also it will be in this phase is where we characterize and validate the engineered compounds that we are building up. I need to say that transversally, we will have to be sure that all the process is sustainable, and this is responsibilities in work package 7 and work package 6 that are in charge of the sustainability through life cycle assessment, considering attributional and consequentiality approaches, and will be in charge of identify key aspects to guide the process development and compare the products and processes to counterparts that to current counterparts that are basically the synthetic surfactants. Finally, only the processes that are being able to be upscaled, they will be evaluated in their encapsulation performance and the activity as delivery agents of RNA interference gene therapy, their anti-tumor activity that could be potentiated, or they can be drug delivery agents of anti-tumor molecules that are very insoluble, and their application of siderophores as antimicrobial and antifungal compounds, especially favoring the formulations of new antibiotic siderophore conjugates in the so-known Trojan horse strategy. So the main impacts of our project is supporting the European Union in becoming global leader in biosurfactant industry. So through the synthesis of tailor-made compounds according to the industry and consumer needs, and pushing forward to an European greener technological industry by combining powerful synthetic and system biology tools. So the idea is that we are also developing highly efficient microbial platforms. We are reutilizing microbial collections. We are reutilizing all available information in the databases. We are not overexploiting their natural resources by extensive and expensive screening campaigns. We are reutilizing everything that is totally aligned with the circular economy concept. So the development of highly efficient microbial platforms also remarks the importance of protecting the oceans as our source of biological diversity. In this way, altogether, we aim to connect the blue and white biotechnology by using these molecules that they can organize in supramolecular architectures, and they can be used in quite a lot diverse fields of applications that as cosmetics, household care, from industry nutrition, agrochemical industry, and pharmaceutical applications, particularly the case of genetic material delivery. So, any questions? Thank you so much.